Hello, everyone. Thank you for joining us today for the Molecular Connectivity Online Series, or MCOS. My name is Cindy from New Jersey Institute of Technology from the US. For those who come for the first time, the Molecular Connectivity Working Group is a community of researchers dedicated to promote molecular imaging as a tool to study brain connectivity. We plan this online series as a platform to promote and exchange ideas of latest research in this field. You can visit our website, subscribe to our newsletters, or follow us on Twitter or LinkedIn. So before we begin, a quick note. We are recording this session and will upload it into our YouTube channel. To protect our speakers' research, we kindly ask you to not privately recording this and upload it to any third-party platforms. So now I'm pleased to introduce today's speakers, Dr. Paul Klauser and Dr. Federico Lucchetti. Dr. Klauser is a child psychiatrist and assistant professor at the University of Lausanne, where he heads the research unit on psychosis. Joining him is Dr. Lucchetti, a physicist and a PhD in neuroscience. After specializing in resilience at the University of Luxembourg, he joined Dr. Klauser's group in 2023 to focus on whole brain metabolic connectomics. Today, they are presenting on constructing the human brain metabolic connectome with MR spectroscopy imaging, revealed cerebral biomechanical organizations. Thank you again for joining us, and please join me to welcome Dr. Klauser and Dr. Lucchetti.
Thank you very much for the invitation. I will share my screen. Cool. So, good morning, everyone. So, I'm Paul Klauser and my colleague Federico. So, I'm going to start with the general introduction about our research interest on early intervention in psychosis. I will also provide with a short introduction about MR spectroscopic imaging. And then my colleague Federico will continue with the construction of the metabolic connectome. So regarding the context, I think it is now well accepted that psychotic disorders are best described in terms of clinical stages. It starts, this clinical staging model allows to follow indicators of an underlying pathophysiological mechanism. So it starts with the premorbid stage, during which adolescents from the general population they experience non-specific symptoms such as attenuated anxiety or depression. Then you have the better known stage called clinical high risk or at-risk mental state for psychosis, during which young people experience subclinical psychotic symptoms, for example, hallucinations, but with intact insight. So they recognize that something wrong is going on. Unfortunately, we know that 1/3 of those adolescents or those young people will experience a first episode psychosis within the following 3 years. This first episode psychosis stage corresponds more or less to when we usually start prescribing antipsychotics. And unfortunately, we know that 2/3 of those young people experiencing a first episode psychosis will experience long-term psychosis such as chronic schizophrenia with chronic or recurrent psychotic symptoms. So I think with this clinical staging model in mind, it's easy to understand that the challenge for both clinicians and scientists are first to be able to identify young people at risk as early as possible, and also to develop interventions, novel interventions, but which are stage specific, because of course you won't provide the same intervention to young adolescents from the general population as so it's going to be different to the intervention that you're going to provide to older adults, for example, with chronic schizophrenia. So basically, we have two aims. So first, we try to identify biomarkers, for example, diagnosis biomarkers to improve early diagnosis. And we also try to uncover novel targets that could be targeted by novel treatment, novel intervention. So for that, we do translational research that has started more than 20 years ago with Kim Do and Philip Koenig. You may know them. And basically, this translational work rely on both preclinical research based on animal models and also clinical data. We have cohorts of patients at different stage of psychosis, at risk, first episode, chronic, and also, of course, matched healthy controls. And all these participants, they are, of course, assessed from a clinical point of view, but they also go through neuropsychological testing. They get brain MRI, they get EEG, they give blood, and we also perform a skin biopsy to get fibroblasts that we can put in culture. So, as I told you, we have basically two aims: early detection and stage-specific intervention. So regarding early detection, so you retrieve here the first stages of psychosis from left to right, so the premorbid stage, prodromal, and early psychosis stage. And at the bottom of the slide, a summary of our working hypothesis. So a vicious circle between excessive stress and neuroinflammation that leads to alterations in both white and gray matter, and eventually leading to the expression of psychotic symptoms and cognitive impairments. So our research is based on several techniques. We use IPS technology to have brain cells from participants, so patients and controls in culture. We also try to identify blood-based biomarkers related to redox homeostasis or oxidative stress in the blood, or also markers associated with inflammation. And of course, and that's the subject of today, we also use neuroimaging tools, mostly MRI, so structural MRI, functional MRI, diffusion MRI. And more recently, we have been interested into whole brain 3D MR spectroscopic imaging, or MRSI, because we think it's going to be more sensitive to detect the early and subtle brain alterations in psychosis. You may know that brain alterations in psychosis, if you do group comparisons, they are obvious in the more advanced stages of psychosis, but they are much more difficult to highlight in the earliest stages. Just a couple of words, it's not the subject of the talk today, but we also work on developing stage-specific interventions. So here you retrieve the three first stages of psychosis. Just to give you an example, so 10 years ago, we completed a clinical trial on NAC, that is an antioxidant, that was administered in first episode psychosis patients. A couple of years ago, we completed a clinical trial on mindfulness training in young adolescents from the general population. And currently, we are running a translational project on gamma neurosensory stimulations, so visual and auditory stimulations at 40 Hz trying to restore cognitive impairments in those at risk for psychosis. So coming back to trying to improve early detection using neuroimaging tools. So I think you will agree that there is a huge gap between the findings from most of the currently used neuroimaging tools like structural or diffusion MRI and the findings from post-mortem studies using microscopy. And so we have the impression that there is really something missing in between to bridge the gap between micro and mesoscopic findings. So MR spectroscopy is maybe a good candidate to bridge this gap because with MR spectroscopy, you can quantify metabolites in a volume of interest. MRSI is probably better because you can not only quantify the metabolites, but also assess their spatial distribution. Unfortunately, this classical MRSI technique is limited to a rectangle in the center of the brain. The resolution is coarse and the acquisition time is really long. I think, if I'm not wrong, it's between 45 and 60 minutes for the acquisition. So that's why 15 years ago already, colleagues started whole brain 3D MRSI. However, this approach had a low spatial resolution and also suffered from a long acquisition time and also a very low signal for GLX and inositol. So GLX is glutamate and glutamine. That's why more recently improvements were made using fast ultra echo time whole brain MRSI. So it's an echo time under 1 millisecond that drastically improve, of course, the acquisition time, but also the signal for GLX and inositol. However, this technique was still suffering from a lot of noise. So this is for this reason that we collaborated with Antoine Klauser at the CIBM in Geneva. And Antoine worked on a novel fast whole brain high resolution 3D MRSI acquisition and reconstruction technique. So first in 2 dimensions, but also in 3 dimensions at the 3T scanner. So with a resolution of 5 millimeter isotropic and with an acquisition time that is now under 12 minutes. So you can see here the metabolic maps of 5 groups of metabolites: NAA, creatine, choline, inositol, and GLX. And more recently, the technique was also developed for the 70 MRI scanners. The advantage with the 70, you have a better resolution, 3 millimeter isotropic. You can also differentiate glutamate from glutamine, and you can get GABA. So that means that's very exciting that basically at the 70, you can map glutamate and GABA. So basically you can map, you can have an index of the excitation on inhibition ratio. So what's the place of 3D MRSI in neuroimaging? It seems for us that's a bit of a hybrid approach because it provides, of course, information regarding the metabolic content, but it also provides information on brain structure. I mean, we can recognize brain structure from these metabolic maps. So it's static, it's not dynamic. The acquisition time is more or less equivalent to what you would get for a single voxel MRS. But of course, you have a spatial resolution that is much better. You can map the entire brain and with a resolution that is more or less comparable to what you would get with functional MRI. I told you between 3 and 5 millimeter isotropic. So there is growing interest in 3D MRSI, not only for research in psychiatry, but also for research in neurology. I mean, there are 3 main reasons for why it's not more commonly used. First, it's still a research prototype. Even if the WIP is going to be probably released soon, it's still a prototype. That means also that you have to do the reconstruction offline. So the reconstruction of the map is not performed at the scanner. And also there was no pre-processing tools except the one that have been developed by Federico. And we'll talk about it later. So in Lausanne, we have implemented this technique a couple of years ago already, specifically in those at risk for psychosis, because we believe that it should allow us to detect the early and subtle brain alterations before a first psychotic episode.
All right. Thank you, Paul, for this nice introduction. I will now take over and explain to you how to generate this fancy image about the brain metabolic connectome as measured by MRSI technique. So as Paul already introduced to you, there is a growing gap between linking cellular neurobiology and mesoscopic and macroscopic imaging as performed by MRI or PET scanning techniques. Of course, we can always increase your spatial resolution, maybe temporal resolution to get even closer to neurobiology until you hit the barrier of physics where you can't excite enough volume, enough atoms in a smaller volume. But I think another question would be more interesting is that are we missing maybe a new dimension? That new dimension would be called frequency here in the context of MRI, or maybe chemistry, neurobiology. Imagine going to an audiologist and for a hearing screening, and the audiologist only asks you whether you hear or not. I mean, it's a useful question, but it's not going to lay out what the underlying biology is. Rather, the audiologist is going to ask you at what particular frequency are you not hearing? And based on the response, whether you're losing hair cells in the higher frequency spectrum or the lower frequency spectrum, we can pinpoint exactly where on the basilar membrane in the cochlea your hearing loss is localized. And so the missing dimension here was definitely frequency. Equivalently, in MRI, once you suppress the predominant lipid and water signal that you usually get in structural images or bold images, you're left out with this spectrum, the MRI spectrum here on the right. So on the x-axis, the frequency, every time you see a peak that corresponds to the signal amplitude to a particular metabolite. Here what you see is the creatine, myoinositol, choline, GLX, and tNAA. The height of each peak corresponds to the amplitude of that particular metabolite to the global signal. Now this is single voxel. We can do this on the entire brain. Now at 3 Tesla with 5 mm isotropic resolution, about 12 minutes acquisition time, and we can scan 5 metabolites with one single go. Not each one by one, but it's a multi-metabolite image. So this is what the images look like. I'm just showing you a few slices so you know what to expect. And then the question is, I mean, what do these signals mean? Well, we know more or less what they mean. They are mostly um, precursors to very well-known neurotransmitters, like, for example, glutamine, glutamate, which is the common excitatory neurotransmitter that we all know. What about choline, acetyl aspartate? They're all somehow involved in cellular mechanisms, energy metabolism, glial cells proliferation, etc. No matter what we know about them, what we know for sure is that they're somehow involved in some complex, intricated metabolic network. And because we have access to these maps for the entire brain volume, why not compute some kind of network, a metabolic network that would quantify how these metabolites interact across brain regions? And that's exactly what we did. So why doing all of this? As Paul already mentioned to you, that the first thing would be to build some foundational knowledge and then maybe use that foundational knowledge to find a biomarker for early onset of psychosis and maybe, find metabolic network disruptions pretty early in disease progression, ideally before structural and functional changes appear. Okay, let's get right into it. For a network, we need nodes, we need edges. All right, so how do we do this in in other modalities? So the first one, structural, the dimension that we are sampling is definitely space. The domain is we are looking at diffusion water molecules. The nodes are mainly static gray matter nodes, and the edges correspond to fiber tracts. So the more diffusion you get, the more dense your fiber will be between two gray matter nodes. For functional, it's a bit different. The dimension that we are resolving is temporal. The domain that we are looking at is bold signal. For PET, for functional PET, it's not that different actually. We're looking at glycodynamics. So instead of looking at neurovascular coupling, we're looking at how glucose uptake varies across the brain. Nodes here are also time resolved, so dynamical gray matter regions. and the edges just corresponds to a correlated activity, whether it be a bold signal or a glucose uptake signal. For metabolic, this is again different. We don't resolve in time. We only have access to spatial dimension. The domain here is not diffusion, not bold, no glycodynamics. The domain here is actually metabolite signal because we have those 5 maps. So per region, we have 5 different values. Those 5 different values, we can cross-correlate them with all the other nodes in the network and to form something called a biochemical similarity or biochemical correlation across the brain. And that's exactly what we did. The way you do it is you postulate your gray matter regions with your favorite atlas. You map out the 5 values inside every parcel. Just take the median, the average, whatever you like. And then you cross-correlate all of these different regions to get this matrix. Okay. It looks very familiar maybe to you, for those of you who are doing functional connectivity. If you look closer, again, you see those big red. So we can do this within subject. This is the new thing. This is not a cross covariance matrix. We can acquire one single matrix per subject, or maybe even compute an average across a group of healthy individuals. We did this for 52 adolescents. And if you look closely at this matrix, so the red means positively correlated, blue means negative correlated. The red regions, those are the neocortical hemispheres that appear. The smaller ones are the subcortical regions, and the bottom far right corner is we see a small part of cerebellum, not the entire thing because the coverage wasn't that good at that particular region. Okay, once you have those matrices, we can run the same usual statistical techniques. For example, recruit 2 different kind of cohorts, healthy controls, and maybe for our interests, 25 patients that were at risk for psychosis and run the usual network-based statistics from Zelinsky 2010, and then see whether there are disruptions in the network. And we can see disruptions in the network, but we have absolutely no idea what they mean. And so the next slides are going to be dedicated to explain to you what does this network actually mean in order to make sense from this statistical analysis. Okay, I'm going to use different tools that researchers have used in different other modalities. The first thing that needs to be done is how do these correlations distribute over space? What you see is the closer a region is, so on your x-axis, you're on the right plot, the closer they are, the more positively correlated they're going to be. This makes sense because nearby region will have the same metabolic profile. The further they are away from each other, the more dissimilar they are in a metabolic space. Okay, this makes complete sense. Nothing unexpected here. Let's see what the first component of, for example, a t-SNE or a diffusion embedding would be. So the first component, meaning we're going to squeeze this matrix down to one-dimensional array and look at what's the first component that explains the most of the variance or the variability in our data set. We do this and then we project it back to our 3D brain to construct something that we call the first metabolic similarity mode. I mean, people usually call this gradient. Now I like to call it the mode because there's no gradient involved. But I will explain it to you later. So the first pattern that you see, again, we are restoring this proximity rule, is that 2 regions that are close to each other will have the same color. So the same color means here the same metabolic similarity index or mode. The closer they are, the same gradient color they will have. The further they are away, the more anti-similar they are, with a few exceptions. If you look closer, so for example, between you get a nice separation between your sensory and motor cortex. They're close to each other, but metabolically very dissimilar. And this allows me to formulate the first principle of MRSI connectomics, meaning that any 2 regions that are close to each other will share the same metabolic similarity or minimize local variation. But this pattern that we outline is clearly not sufficient to explain all the variants. And we can run, let's say, 10,000 geometric models that respect these rules and try to correlate with empirical data. But you see, the best one only explains 5% of the empirical data. So this first principle is clearly not enough. We need a second one, maybe a third one. Not sure. All right. Before we get to the second principle, the other observation, striking observation that you can do is we get nice separation between regions that we knew were functionally and structurally different. For example, you see a separation between sensorial and motor cortex, between higher order and motor sensory or unimodal and transmodal. You get a nice isolation of the limbic system and cortical versus subcortical separation. And I mean, what's the new thing here? We already knew that those were separate. Well, the new thing here is that we are separating these regions not based on structural or functional criteria, but based on biochemistry, right? Because we have access to these 5 metabolites, we can exactly find out what are the metabolic profiles that are going to separate these regions from each other. And so when you map those 5 metabolites, so the tNAA, inositol, choline, GLX, and creatine back to this metabolic similarity mode, what you see here is, so the violet regions, so the sensory visual cortex will have a lot of tNAA and very few choline. And this pattern is flipped the more rostral you're going to move. So in the frontal regions, you get an inversion of this pattern also in the subcortical region. So the driving factor here is mainly tNAA and choline ratio. So this ratio exactly is going to explain to you why these regions are metabolically and then later on also functionally different from each other. Let's get maybe to the second pattern. If you do an histogram of this metabolic similarity mode, what you see is it's almost flat. Almost. I mean, you could compare it to random uniform distribution and see, and for example, compute entropy, and you will see it's pretty high compared, it's very close to random uniform distribution. And this allows me to formulate the second principle, meaning that your network tends to maximize entropy. You can observe this pattern in all of the individuals, not just the average. All the individuals that we scanned across cohorts, these two principles are always optimized against each other. So why am I telling you all of this? It looks very nice, but what's the use of it? So imagine just a very, very simple example. Take a 1D grid here, an empty 1D grid. Now let's enforce the first principle, the geometric principle, meaning that proximal regions will express the same metabolic profile. What you get is everybody will look the same to each other. Very boring, nothing interesting here. If you go to the other side of the optimization is that let's distribute those values as uniform, as random as possible. Also, nothing interesting here. Just everybody will have a different color or a different metabolic profile. Now, what happens if you take the first geometric, the green line in the beginning, and just add some randomness to it, but still keep some of the local variance enforced? You will get something interesting happening. So you will have 2 gradients, 2 vectors appearing, 2 variations appearing, right? Just by adding some noise to it. And if you're going to have to find a middle line, a good balance between minimizing local variance and maximizing entropy, you will end up with a pattern that looks like this. You will get a monotonic increase or decrease of those mode values, forming a single gradient across the network. I mean, it works well for 1D. For 3-dimensional data, like brain data, it still works, but it's just a little bit more complicated because you get a lot of different things going on at the same time. And that's exactly what you find. We see different gradient patterns appearing throughout the brain. And the most common one that you can see is the gradient that progresses from posterior sensory regions to your frontal lobe regions, ending up somewhere in the subcortical regions. And this progression is mainly explained by the ratio of tNAA over choline. So you get a smooth variation of those two metabolites is going to drive this posterior to anterior gradient formation. And when you map this out into a 3D brain, that's the fancy image that you can generate. I mean, we saw those two principles also playing out in different modalities. So for example, in structural brain networks, or what has been called the economy of brain networks, the first principle would be equivalent to minimizing wiring cost, right? So you're only going to wire regions that are close to each other and keep the long range connections as sparse as possible to maximize efficiency. This is exactly what we see, but in a node-based network, not in an edge-based network, what we call the economy of metabolite distribution. This gradient analysis is nothing new. I mean, it has been performed in so many different MRI and PET modalities. The one that comes closest to what we see in MRSI is the one here in the red square, where researchers have effectively merged genetic data with structural data, and they can see the same kind of gradient appearing, so the rostral to anterior gradient that they think was mainly driven by thalamic orchestration. How does these networks, metabolic networks, as measured by MRSI, compare to other known ones? So structural connectivity, very poor correlations. So a structural edge doesn't mean that things are metabolically similar to each other. It's only 0.05, the best correlation that we got. For cytoarchitecture, it's a bit better. So we get a almost 0.3 correlation with the big brain cytoarchitectonic data sets. For genetic co-expression, it's even better. We get a 0.46 with the Allen Brain Atlas, where we computed similarity based on genetic profile expressions. So this concludes the significance, the interpretation of these networks, that they have a very strong foundational biological meaning, and they're not just, artifact of our method. Let's get back to our group comparison result that I showed you in the beginning. So again, we recruited 25 healthy controls, 26 patients that were at risk for psychosis. And when we order all of these nodes in the network along this metabolic similarity mode, meaning that we are ordering them so as to respect this monotonicity, right? So we're starting in violet, going to orange, going to green, going to blue again. What we see here, the circles are the healthy controls. They stack up very nicely around that circle. But if we overlay the patients in squares, you suddenly see that there is a disruption of that pattern. And we can localize the disruption more or less. It's pretty diffuse, but we can find out where exactly this pattern is broken. And it turns out that the regions where this pattern is broken, this is exactly where the NBS statistics found a disruption of the network. So this is very interesting. So not only did we know that similarity is somehow disrupted in patients at risk for psychosis, but we can explain that this decoherence in the first metabolic similarity mode that we observed was mainly due, to a disorganization of this pattern. And we can explain this disruption now, for example, in looking what tNAA and choline levels were at that particular regions. And what we saw is that choline was maybe too high in the prefrontal cortex and NAA, acetyl aspartate, was too low in the thalamus. Conclusions. So we did this analysis also in structural connectivity, and we found no results. Far away from that. we found results in metabolic similarity, I mean, which already proves for the little sample size that we have, about 51 uh, uh, individuals, we already found some nice results that explains the difference between the groups. And we can explain this loss of metabolic similarity not as a pure artifact of statistical testing, but we can tie it to a decoherence or of abnormal widespread metabolic change. Um, Lastly, if you're interested in the papers and all the methods that we developed, just look at these two papers. So the first one published in Nature Communication is all about this connectomics work. The second one, published by one of our colleagues, Edgar Celereau, used mainly a voxel-based analysis to find differences between groups. Uh, Along these years, we have developed a whole pipeline of MRSI data pre-processing, normalization, T1 registration that you can freely download. The code is on GitHub. It's maintained every day. And we just recently published also on GitHub a second software tool that enables to do this connectomics analysis on the similarity matrices. And with that, I want to thank Paul Klauser's research unit for psychosis, all our colleagues and collaborators from Patrick Ackman Connectomics Lab. And with all of this, I'll thank you for your attention, and Paul and I are happy to take your questions.
Thank you very much. Really a wonderful talk. Anyone have questions, feel free to unmute yourself and just to ask. Ariana?
Hello. Thanks a lot for the talk. I have a few questions. Like, I always wonder when I see this kind of, let's say, spatial similarity-based approaches, like, what's the rationale for considering them as connectivity? I know that a few papers have come out using these approaches both in structural MRI, in PET, and now you did the same. So I was just wondering, like, if you can explain, why going for that? Why not going for an intersubject approach? And I also wonder if with this technique, you don't have a chance to do a longer acquisition to have a sort of time series of some kind, or yeah, if you can explain
Yeah.
a little bit more. Yes.
The connectivity part, I agree with you. We shouldn't call this connectivity, nor should functional connectivity be called connectivity. I mean, we shouldn't get hung up with those terminology. I think the thing that we are really measuring here is some kind of network that is based on some higher dimensional manifold. And this is the whole, I don't know if you're following the gradients, the cortical gradients community. I think this is much more useful framework to understand what is happening here. And as you can see, it really gives you nice interpretation on how to interpret these findings. Um, why intersubject, I think was the second questions. I mean, I think it's always more interesting to have a within subject measure because then we can, actually have a clinical application for it. I mean, intersubject would be interesting to have something like a population average study, but within subject is more attractive for people that are going to dedicate themselves to translational studies. Your third question about to get multiple scans, or maybe yeah. I mean, we're still at 12 minutes, 7 minutes for the 7 Tesla. I have heard rumors that people are trying to achieve maybe single slice in 1 minute resolution. I heard different rumors that people are now playing, I think it's in Geneva, to have 3D on the 70 time resolved at 1 minute, but I'm not sure whether they will be able to achieve this. Maybe a few results are going to come out next year, but it's still very speculative at this moment. We are really at the cutting edge right now of acquisition times.
Hey, can I ask a quick question?
Yep.
First of all, thank you so much for the talk. Such beautiful data. The question I have, and I'm sorry if I missed this, so you might have included, have you done any test retest on this measure in the same subjects? And if yes, what does that look like?
We only replicated it in different cohorts in let me think, 3 or 4 different cohorts with 3 or 4 different scanners, and we get overall the same replicable pattern. On the same subject, I think only on myself 3 times or 4 times, and it looks mostly the same. Yeah.
Yeah. So when they developed the technique, they did it. You can find the information in the seminal paper. It's just that, so of course, I mean, it depends on the group of metabolites, but it's within the expected range of a couple of % of difference between a scan and rescan. So it's comparable to what you would get with a single voxel spectroscopy.
Better than PET. Thank you.
Can I ask another question? I liked really a lot of slides where you show the different applications for whole brain, for different metabolites. But I was wondering, like, so can you get a whole brain image of glutamate and GABA at the same time? So whole
Yes.
brain, including
At 7
cerebellum,
Tesla, yes.
with 7 Tesla. Okay.
Including the cerebellum. We've done tests, so but you need the 7 Tesla, as Federico mentioned, and you can really have I mean, virtually nearly the whole brain. I mean, there are some regions, for example, the orbitofrontal regions, because they are very close to the sinuses containing air. So yeah, there are some regions for which it's more difficult to get good signals. So I would say virtually whole brain, but not exactly. But you can have the brain and the cerebellum, yeah, and glutamate and GABA at the same time. And as you mentioned, that's very exciting to be able to map these two yeah, in the same people at the same time.
Okay, so I have some questions. So firstly, so you have this spatial distribution, like say the regional variations of this metabolite, and also you have this constructed this metabolic matrices, and then when you show this similarity with others, with anatomical connectivity or the genetic maps, so which one did you use? So when you compare this similarity, when you use the matrices and see how these matrices are similar to those, but have you also correlated the similarity with the regional variations and how they are similar or not, and how to interpret this?
Yes, I didn't show the results here, but they're somewhere on the paper where mainly correlating region-wise things. I, th- I don't remember exactly, but where we got the best kind of correlation across cytoarchitecture and genetic information was somewhere maybe in the limbic system and the motor cortex. I'm not sure about it. I have to cross-check this. But for the matrix correlation, we just correlated edgewise and then corrected
Yes.
for
Right.
spatial autocorrelation and so on.
Okay. So for furthermore, you compare these patients with healthy controls. So have you compared the regional first, and then did you see regional differences, and then whether that drives this differences in the similarity matrices differences?
I took a very agnostic approach, so we didn't have any initial hypothesis of where the disruption might be. Also, it's a network, so everything is kind of intricated. But the regions that were lightening up the most in the comparison was the thalamus, the prefrontal cortex, and the motor areas to some degree.
Okay.
As I showed you, I think was one of the other slides. Yeah.
Right.
So here are the regions that, where you saw the most disruptions happening. And I mean, the regions that were happening, the most recurrent across subjects was the prefrontal cortex and the thalamus regions.
Yeah. So yeah, I'm not a biochemist. So the question is that what you measure here, is that something more trait-like or is state? Like if you measure several times, do you expect that they are similar? Or if you measure like over time, whether you see some variations,
Yeah.
and if you could do that, does that really mean something? It's like what Scherner has done, the metabolic, glucose metabolic, you can do with functional PET, you can correlate that signal, whether it would make sense
Yeah.
here.
There are people doing functional MRS,
Ah,
so meaning time-resolved MRS, but it's single voxel.
yeah, okay.
And the metabolite that varies the most inside, so at the minute resolution is glutamate and glutamine, right? The excitatory neurotransmitter. Choline and tNAA is mostly static over days, I think, or even over months. Um, but I remember, I think it was inositol or creatine. No, creatine is still very static. I think it's mostly glutamate and I-glutamine that are going to vary depending on the task that you're subjected to.
Thank you. Yeah,
But that I think that's a very pertinent question. I mean, is it a state or a trait that we measure here? And I think that's still a relatively open question.
thank you. Anyone, if you have questions, feel free to unmute and just ask.
Just one more thing I thought about. I mean, what can we tell you is that all the participants have been scanned at the same time of the day. So it was always early afternoon. So
Yeah.
at least if there are, variations during the day, they all have been scanned at the same time of the day,
Yeah, yeah.
more or less.
A question for noobs. Essentially, like, how do you have to normalize your data to some extent? Like, is there any scaling applied? Do you get like an absolute measurement that reflect the actual quantity of glutamate, like in absolute term, or how does it work?
They are not absolute units. They are, what do they call them? Institutional units. So it's not calibrated, if that's the question.
All right. And so, like, I don't know if you check for, is there any effect of, is there different ways to do this scaling to make the data usable, like to quantify
I
them?
mean,
And does this has an impact on, yeah?
there are different MRSI techniques, and everybody uses a different kind of approach. I mean, usually people are referencing it to the water signal, for example, or to the
Right.
creatine signal because it's mostly completely uniform across the brain. So it gives you a very stable gauge from where to look at. But again, these measures, they are not calibrated, but they are relatively calibrated to each other. And what is very attractive here for the connectomics part is that we don't care whether they are calibrated because we are going to cross-correlate with respect to each other. So a global scaling factor wouldn't affect our results. I mean, even for voxel-based analysis, I don't think you will be sensible to this. I mean, Edgar, so the other paper that was in preprint, he showed that he was able to replicate his results in two different cohorts, in two different scanners, but the same acquisition sequence.
Very interesting, thanks.
So do you know that there are, do you plan to make your data public available, or do you know there are any data sets that are like MRSI data sets that we can try to look at?
The MRSI sequence will be made public
Right.
very soon. I think it's a work package that you can download if you have a Siemens scanner.
Yeah.
The data we are looking into it to maybe share at least all the derivatives, so the connectivity matrices, for example.
Right. Yeah.
For the other, I think for the raw data set would be difficult because it's clinical data.
Right.
We're not sure if we can share this, but of course, I mean, this will, in the next coming months, we'll have a discussion
Right.
and try to find a way to make this public.
Thank you. Um, does anyone have other questions? If no, I think it's about time. So we are now quite, about 10 a.m. here in the US. Thank you very much again for giving us a very wonderful talk and very wonderful data results. And thank you again.
Thank you. Thank you very much for giving
Yeah.
us the opportunity to present this work. And
Right.
thank you everyone for attending.
Yeah.
Yes,
Thank you. Thank you.
Thank you for attending.
Thank you. Thank you. Bye bye. See you, everyone.
Bye bye.